Welcome back to the physiology of the gastrointestinal tract. In the first part of the lectures, I talked about general physiology of the gut. I gave it a definition. I also talked about the control system that orchestrates the various functions of the gut, intrinsic and extrinsic control systems, both of which contain nerves and endocrine secretions. I then talked about the special functions of the gut in the second part of these lectures, which is special physiology. I talked about the very first function in the gut, and that is motility. I've mentioned that the smooth muscle cells are the main contractile tissue in the gut. I also mentioned that these smooth muscle cells are innervated by enteric neurons that contains vercosities. I also mentioned that the smooth muscle cells themselves communicate with each other through nexuses or gap junctions, which allows relatively free movement of ions between these cells. Then I mentioned that you have two types of contraction in the smooth muscle cells. One is phasic, which means it stays for a shorter period of time, and one is tonic, which lasts for a relatively long period of time. It can reach hours. We talked about the mechanism for contraction, that we need to change the action potential of the membrane of the smooth muscle cell, followed by increase in the intracellular calcium content. This will lead to interaction between actin and myosin, which will lead to muscle contraction. After that, I mentioned that we have to have two requirements for the smooth muscle cells to contract. One requirement is an action potential coming from an enteric neuron. Second, we have to have a slow phase coming from the interstitial cell of Cajal, or the ICCs, especially, as I mentioned, the plateau phase of that wave. With these two requirements, contraction can happen. I moved and started talking about the specific motility patterns for each part of the gastrointestinal tract. First, we've divided the gastrointestinal tract into four parts: the oral cavity, the esophagus, the stomach, and the intestine, both small and large. And in the previous lecture, I talked about the motility patterns in the oral cavity, in the esophagus, and in the stomach. These are the four parts of the gastrointestinal tract that we started talking about their specific motility patterns. We've mentioned that the oral phase has two stages: an oral stage and a pharyngeal stage. The oral stage digests the food partially, increases the surface area of the food particle, so digestive enzymes in the oral cavity, such as lipase and amylase, as well as the saliva, can mix with it so it can go to the next stage, which is the pharyngeal stage. The pharyngeal stage has no digestion. It only involves motility, swallowing. I said that the oral stage is under the control of the mandibular and the trigeminal nerves, whereas the pharyngeal stage is under the control of the glossopharyngeal and the vagus nerves. And I said that this phase, which is the oral phase, fails when the person or the animal suffers from cerebrovascular injuries, such as strokes. Then I moved and talked about the specific motility patterns in the esophagus. And I said, also there is no digestion, just as the pharyngeal stage of the oral phase. The other important thing about the esophagus is that there is no ICCs, meaning that there is no slow waves, which means that the contractions are myogenic, which means that the smooth muscles of the esophagus are able to generate contractions on their own. They can contract, and the basic motility pattern in the esophagus is a primary peristaltic movement. Sometimes the primary peristaltic movement fails, therefore a secondary phase of these peristaltic movements take place. We call that secondary peristaltic movement. Again, there is no digestion, but with this motility patterns, there is dilation or relaxation of the stomach to accommodate the food coming from the esophagus. Again, no digestion here. The stomach, the third part of the gastrointestinal tract, contains two motility patterns. One is contraction, the other is relaxation. In the contraction motility patterns, we have two motility patterns. We have a reservoir which has tonic activity. That means the contractions continue for a long period of time, so the food will be broken into very, very small pieces, and it goes after that to an antral pump, which basically has a phasic type contraction that lasts seconds. This motility pattern leads to the second motility pattern in the stomach, and that is the relaxation in the stomach. We call this motility pattern, the relaxation, gastric emptying. Gastric emptying allows food particles that are 1 millimeter in diameter to move or less, of course, to move into the upper portion of the duodenum. This happens under both enteric and hormonal control. The hormone that regulates this gastric emptying is cholecystokinin, whereas the contractions in the stomach, the reservoir, and the antral pump are regulated by two innervations, the vagus and the enteric nervous system. The vagus and the enteric nervous system. In this lecture, what I will do is I will continue talking about the specific motility patterns in the gut, and I will talk about the intestinal motility patterns. The motility patterns in the small, followed by the motility pattern in the large intestines. However, before I start talking about these motility patterns, I would like to draw your attention to the fact that the motility patterns in the small and large intestines depend on two things. The two things that the motility patterns of the small and large intestines depend on are the digestive and the interdigestive states of the gut. What do I mean by that? I mean that when the animal or the human is actively involved in eating, it has motility patterns that are different from a person or an animal that is not eating. Or you can consider this fasting. We don't mean it in the literal term, fasting, but what we mean by that is the fact that the person or the animal are not actively engaged in eating as we speak. This is the interdigestive state. The digestive state is when the animal or the person is actively engaged in eating as we speak, now, at this second, at this moment. Interdigestive state is not. After I talk about the motility patterns of the gut in the small intestines and in the large intestines, I will talk about the clinical relevance of these motility patterns in the small and in the large intestine. I mentioned that the motility pattern in the small and large intestine depends on the state of the gut. I said that we have two states in the gut. One is when the animal or the human is actively engaged in eating as we speak at the moment. at the time of recording from the gut, and the other state is when the animal or the human is fasting, is not actively engaged in eating. We call that the interdigestive state. Again, when the animal is not involved or engaged in eating. When the animal or the human is engaged in eating at the time of the recording, then we call that the digestive state. And each one has its own characteristics. For example, when the animal or the person is not actively involved in eating, the hormone motilin is being secreted in the gut. And when the hormone motilin is secreted, it will cause what we talked about last time, and that is the MMC formation, or the migrating motor complex. We will talk about that in a second. But the interdigestive state is when the animal or the human are not actively engaged in eating as we speak, then the hormone motilin starts to be secreted, and that's what will stimulate the migrating motor complex to be generated in the small intestines. In the digestive states or state, that's when we are involved in eating as we speak. We have no motilin, we have no MMCs, but we have food in our gut. So what I will do is I will explain to you the different motility patterns for each of these gut states. First, the interdigestive state. Second, we will talk about the digestive state. So, when there is no active engagement in eating, we have three motility patterns in the small intestines. MMC is the major one. We then have segmentation, and then we have power propulsion to basically throw the food into the large intestine. This is the last or the third stage. But the major motility pattern is the MMC in the small intestine in the interdigestive state. And all of these are controlled by enteric as well as extrinsic innervation, but the majority is enteric control. The enteric nervous system is in control at this stage. Let's talk about the MMC again, just as a reminder. We've talked about it when I've talked about motilin, and now we will just remind ourselves of what is going on with this complex. This is a diagram that shows recordings from the stomach as well as the small intestine, the duodenum, jejunum, and ileum. The MMC is generated by the gut hormone motilin. This MMC consists of three phases: a silent phase, phase 2, and phase 3. A silent phase, phase 2, and phase 3. Phase 2 and phase 3 together. Phase 2 and phase 3 together are referred to as activity front. Activity front. We call the motility pattern between two consecutive activity fronts a physiologic ileus. It's physiologic, it's not pathology. It's not abnormal. This is pretty normal. There is a stricture that happens before the next activity front take place. When the next activity front take place, that's when contraction happens. And the continuous presence of activity fronts, as this diagram is showing, will create what we call a peristaltic movement. A peristaltic movement. So we have three motility patterns in the interdigestive state when we are not involved in eating. The major one is MMC. Of course, as I mentioned before, there is segmentation and there is power propulsion, which propels basically the ingested or the digested food into the large intestine or to the next intestinal level. So what happened in the digestive state when we are actively involved in eating as we speak? What happens when we are actively involved in consuming a meal is three motility patterns will replace the MMC, which is the major motility pattern in the interdigestive state. Now it will be replaced by three motility patterns: mixing, segmentation, and short distant peristalsis. All of these has physiological relevance or physiological importance, and that is to keep mixing the food together to reduce the size to an acceptable size for absorption. Now, it's not the piece of steak that you ate, it is the amino acids that form this piece of steak that the gut needs to absorb so it can distribute to the rest of the body. Remember the definition of the gut? A tube-like structure from the mouth to the anus that does what? That supplies the body, including itself, with nutrients, fluids, and water by performing four functions: digestion, absorption, secretion, and motility. This motility pattern basically is extremely important so the material can be absorbed from the gut to the rest of the body through, again, as we mentioned before, through the portal vein to the liver, to the vena cava, to the heart, to the aorta, again, to distribute it to the rest of the body. Let's see how these motility patterns take place: mixing, segmentation, and short distance peristalsis. This is a picture that shows what happens in each part of the small intestines at the digestive state. These are three segments adjacent to each others. This is what exactly is happening. In the one segment we have mixing, and you have a propulsion in both direction so that the material can be reduced even more in size, and then the end point will be power propulsion so it can go to the a large intestine at that point. So in the digestive state, again, three motility patterns replace the MMC: mixing, segmentation, and short distant peristalsis. Mixing, segmentation, and short distant peristalsis. They replace the MMC that took place in the interdigestive state, the MMC that generated the activity fronts. In the digestive state, this will be replaced by mixing, segmentation, and short distant peristalsis. Now, these are the motility patterns in the small intestines. So, what are some of the problems in the small intestines? Motility related, of course. Well, we have a number of cases. We have diarrhea. We have idiopathic or pseudo obstruction. We have transient ileus. This is post-surgical. We have rapid transit. That's what happened when you have a bad food. And you have IBS. IBS, or irritable bowel syndrome. IBS, irritable bowel syndrome. Very important diseases. Very important diseases. In case of the diarrhea, it will be accompanied by metabolic acidosis and hypokalemia, low potassium, hypokalemia, because the potassium shifts from the extracellular to the intracellular space. So when it's being sequestered in the intracellular space, it goes away from the blood. That's why when you test the blood, it, it will show hypokalemia, low potassium. It's not because you've lost the potassium, it's because the potassium went from the extracellular to the intracellular space. It's being held as a hostage there. Idiopathic or pseudo obstruction, that happens with different explanation. One of them has a genetic component into it. There is no complete understanding of the idiopathic or the pseudo obstruction. Sometimes it can be diagnosed incidentally, and then it can be corrected either medically or sometimes even surgically by doing enterectomy, cutting pieces of the intestine and anastomose them together. Transient ileus post-surgical. It happens because when you have a surgery in the intestine, you will cut the intestine, you will disturb and cut the enteric nerves and sometimes some of the extrinsic innervations to the gut. So when the surgeon anastomose the two pieces together back again, what happens is during this post-surgical stage or the healing stage, what happens is regeneration of these nerves starts to happen again, but it takes time. So that's why you don't have normal movement and normal diameter. That's why you have strictures and you have also ileus as well. IBS, we will talk about it when we talk about the inflammatory bowel disease, which is coming in the large intestine. So now let's talk about the large intestinal motility patterns. It's not gonna be so different from the small intestinal motility patterns. Now we will continue talking about large intestinal motility patterns. You have the same motility patterns are as in the small intestine. However, the intensity of these motility patterns will be different in the large intestine compared to the small intestines because they will accommodate different functions. For example, in the small intestine, you have more digestion and more absorption. In the large intestine, you have more storage and more fermentation and microbial digestion. Again, all depending on the species. In addition to that, we have anatomical considerations in the large intestines that do not take place in the small intestine, and that is the presence of tinea and also haustra. These two structures will be shown in the large intestine of the horse in the next slide. This is a picture of the gastrointestinal tract in the horse. The bottom of the picture shows the left ventral and the left dorsal colons. Left ventral and left dorsal colon. The structure in the center of the picture is the stomach. And to the right of the stomach, the sacculated structure is the small colon. Small colon. This ends with the rectum to the far right of the picture. In the large intestine, you have the tinea and you have the haustra. These are basically contracted smooth muscles that form bands, and also the contraction will form sacculation. The intestine becomes like little sacs. The physiological importance for these structures is that they increase the surface area of the large intestine and they allow microbial digestion to take place. This is a very important physiological function. Now, with regards to the motility patterns in the large intestine, as I mentioned earlier, they are the same as in the small intestine, but with higher intensity because they will accommodate a different function, and that is microbial digestion and also storage. So the large intestinal motility patterns are segmentation, just as the segmentation in the small intestine, but with massive movement. Again, I mentioned that the same motility patterns, but with higher intensity. Higher intensity because they want to accommodate different functions. Propulsion or propulsive contraction. Again, to accommodate the functions which are microbial digestion and storage. And then the last thing, the last motility pattern in the large intestine, and that is defecation. Defecation occurs when the tonic contraction of the external anal sphincter is gone, meaning that there is relaxation in this sphincter. For defecation to take place, you have a massive contraction in the colon, which is the part before the last part of the large intestine. The large intestine comprises of cecum, colon, and rectum. So the colon is the middle part of the large intestine. It gives first a massive contraction movement. The gut contents move to the rectum, and the internal anal sphincter and the external anal sphincter relax, followed by contraction of only the external anal sphincter. This will lead to another milking process of the ingesta to the outside. So first you have a massive contraction in the colon, and the gut contents move to the rectum. After that, you have relaxation of both internal and external anal sphincters, followed by contraction of the external anal sphincter. With this, the ingesta moves to the outside. These are the motility patterns of the large intestine. So segmentation, propulsive contraction, and defecation. Again, similar to the small intestinal motility patterns, however, they have different intensity, higher intensity, because of their function to store food and to have microbial digestion. So what are some of the large intestinal problems? In the large intestines, you may encounter inflammatory bowel disease syndrome, IBD, or diverticulum. May have different times of diarrhea or constipation, diarrhea and constipation. In the constipation, you have decreased motility but increased osmolality. Why is that? Because the ingesta stays in the intestine or in the large intestine for a longer period of time, which leads to absorbing more and more of this ingesta, which makes it hard for it to move in the guts. The Hirschsprung's disease is a congenital disease which is basically a megacolon. Megacolon means enlargement in the colon because there is no enteric nervous system. The Hirschsprung's disease is congenital and therefore it leads to death. It leads to death. Megacolon, on the other hand, can happen at any age, and it can be treated surgically as well as medically. So Hirschsprung's disease is the same as megacolon, but only in newborns. Megacolon happen in all ages, at any age, and it is due to the loss of the enteric nervous system in the colon. That's a very, very important cause. That shows you the importance of the enteric nervous system. If there's no enteric nervous system in one piece of the gut, that piece cannot contract and it becomes like a balloon, swollen and enlarged, and the ingesta in it can become toxic, which leads to death and toxemia, and sometimes septicemia, meaning that the bacteria and the toxin are in the bloodstream. Hirschsprung's disease is pretty fatal. Now, here you have two diseases that needs to be known to you: inflammatory bowel disease, or they call it sometimes inflammatory bowel disease syndrome, and irritable bowel syndrome. The inflammatory bowel diseases, IBD, is Crohn's disease and ulcerative colitis. It's two diseases. The inflammatory bowel diseases, plural, two diseases. Crohn's disease, which happens in the small intestine, and ulcerative colitis, which happens in the colon. In these diseases, I should say, there is inflammation and there are lesions. You can see lesions endoscopically. In case of irritable bowel syndrome, IBS, there will be no lesions. No lesions. It happens in all the intestine. So in that aspect, it is similar to Crohn's disease and ulcerative colitis, but there is no lesions, and the signs which we've talked about before, like the diarrhea, abdominal pain, sometimes constipation, all of these signs, flares, like goes up and down. No time to control it. No time to basically pin it down. So that's why we have to differentiate it from IBD. IBD are two diseases. IBS is one disease. This is a picture of Hirschsprung disease, which is a congenital, which is a swollen colon, as I mentioned before. It's a pretty devastating disease. If it happens to newborn, it's fatal. If it happens megacolon in any age, signs will be swelling of the abdomen, as I mentioned earlier, constipation or diarrhea, failure to gain weight, severe fatigue, and things like that, vomiting. At this point, we've talked about the motility patterns in the gut. We've talked about the motility patterns in the oral cavity, esophagus, stomach, small intestine, and large intestine. The motility patterns in the oral cavity or the oral phase consist of an oral stage and pharyngeal stage. In the esophagus, you have primary peristaltic movement. Sometimes it fails and it becomes secondary peristaltic movement. Comes to the stomach, we have contraction and relaxation. The contraction consists of reservoir and antral pump. The relaxation consists of gastric emptying. We came to the small intestine today, and we've talked about the motility patterns of the small intestines, that they are dependent on the state of the gut, digestive state and interdigestive state. In the interdigestive state, the main motility pattern is the MMCs, or the migrating motor complex, which is generated by the gut hormone motilin. In the digestive state, the MMC is replaced by mixing, segmentation, and short distant peristalsis. In the large intestine, the same motility patterns as the small intestinal motility patterns. However, their intensity are higher because they have to accommodate new functions such as storage and microbial digestion. And so the motility patterns of the large intestine includes segmentation, propulsive contraction, and defecation. Segmentation, propulsive contraction, and defecation. Next time, I will start talking about a special motility pattern, and that special motility pattern is in ruminants. In ruminants. So we will be talking about the motility pattern in the gastrointestinal tract of ruminants.